Hello, and welcome to today's webinar, Gene Synthesis, Process, and Applications. I'm Jamie Murray, Marketing Communications Associate at GENEWIZ, and I'm very pleased to introduce today's presenter, Dr. Krithika Vaidyanathan. In the last decade, Krithika has worked on several diseases, including laryngeal cancer, congenital disorders of glycosylation, and X-linked intellectual disability, using biochemical, molecular biology, and genetic approaches. She has a PhD from the University of Georgia and 2 postdoctoral fellowships at Sanford Burnham and the University of Georgia, both in glycobiology, biochemistry, and molecular biology. At GENEWIZ, Krithika is on the project management team, providing support, project consultation, and communication to our global customers. If you have any questions for Krithika during the webinar, please enter them into the chat, and we will try to address as many as possible at the end of the session. Without further ado, please welcome Dr. Krithika Vaidyanathan.
Today, I will talk a little bit about what GENEWIZ does in terms of gene synthesis, which is a convenient alternative to cloning. The overview of this webinar is as follows. The definition of gene synthesis associated processes and applications, gene synthesis versus traditional cloning, followed by the advantages of gene synthesis provided at GENEWIZ, including special case studies which highlights superiority of GENEWIZ in providing gene synthesis services. Gene synthesis is the de novo synthesis of any DNA sequence. This means that it entails synthesizing a sequence of virtually any length. This synthesized sequence is fully verified by Sanger sequencing, followed by cloning into a PUC57 in-house vector provided by GENEWIZ with both a kanamycin and ampicillin resistance or any custom vector of choice. Also to be noted, GENEWIZ can provide a gene synthesis product as a PCR fragment as long as it is under 2 kb. As an overview of how GENEWIZ proceeds with all gene synthesis orders, the first step is bioinformatics, followed by the second step of oligosynthesis, and a third step of construction of the final plasmid, as well as quality control being the final step. Gene synthesis process entailing bioinformatics is basically the strategy design. So upon receipt of the project design from the end user, we are able to use our in-house bioinformatics tool to design the accurate strategy. This is followed by our team proceeding with oligosynthesis upon confirmation of the project. Once the oligos are synthesized, our team puts these oligos together in a PCR amplification related process. Depending on the size, there may be multiple rounds of PCR as required, followed by fragment assembly to get the larger fragment or the final fragment as seen below. This final assembled fragment is then cloned again into a PUC57 backbone that is provided by GENEWIZ or a custom vector of interest that is provided by the end user. As long as this fragment is under 2 kb and it is not very complex, we are able to provide it as a PCR fragment. The final stages of gene synthesis at GENEWIZ entail quality control. Quality control is a very stringent analysis of the final synthesized product with sequence verification as well as restriction digest analysis. Some of the important gene synthesis applications are listed here. For example, genes that are extremely difficult to synthesize by cloning or ones that lack a template are usually gene synthesized. Groups that work with DNA vaccines, gene therapy vectors, or recombinant antibodies choose gene synthesis as an easier option in terms of turnaround time. Gene synthesis is also used to design predicted genes or different splice variants of one gene, known SNPs, complex genes that have difficult structures such as secondary structures, high or low GC content, etc. This is also an alternative to provide large-scale gene synthesis for microchip-based arrays. Finally, also to optimize codons to boost target protein expression in a particular background of interest. An example shown on this slide is about synthesizing genes that are difficult to clone or ones that lack a template, which brings me to the next point, which is the advantage of gene synthesis versus traditional PCR cloning, as mentioned in the overview. One is the procedure, which directly means that there is a faster turnaround time and a more cost-effective method available. Shown on this slide is a classical PCR cloning process. As you can see, it is a very busy slide, and in reality, the project is also very difficult in terms of having a template and the number of steps that could go wrong wherein you have to go back and start at the beginning. Shown here is the gene synthesis process that you would have to follow when you order a gene synthesis project with GENEWIZ. Here there are 3 steps. The first one is designing the project goals. Second one is to place orders with GENEWIZ. And the third final step is receiving the final product, which is 100% sequence verified, to move on to your experiment. Going back to the gene synthesis application slide, I will talk about another example, which is codon optimization. For those of you that work with protein expression, you know the importance of having the right codons so that the organism you're working with has the right bias for this codon. This is an example of codon usage for arginine by human versus E. coli. Of the 6 codons listed, the first 2 codons are the higher bias for humans, whereas the second 2 are the highest bias for E. coli. This shows that there will be a difference depending on which codon is used for expression in human versus E. coli. This slide is a reiteration of the earlier slide to show you that an unoptimized or non-optimized sequence will follow the trend in the left-hand side, which is the codon usage profile for different codons, whereas when this codon is optimized for expression in a certain organism, in this case E. coli, you can see that only the 90 to 100% distribution is mostly seen. Including the codon usage slides before, some of the considered factors for GENEWIZ gene synthesis with codon optimization is critical elements in the CDS, which include cryptic splice sites, premature poly-A, internal RBS sites. Also of note is sequence difficulties, which are either repeats, both direct and inverted, or secondary structures, as well as GC content. Now I will show a few examples of case studies of the projects that GENEWIZ has handled in the past. It will show you the ability that GENEWIZ has to handle difficult sequences, which include but are not limited to repetitive sequences, which can be both direct repeats or inverted repeats, poly-A regions, high GC or AT content, toxic sequences, unstable sequences, as well as difficult secondary structures. Also of note is GENEWIZ's ability to synthesize extremely large sequences. Case study 1 is about a project where we had multiple repeats that GENEWIZ was able to deliver. As you can see, these are multiple repeats of CAG. As far as the DNA sequence that we synthesize, there is a difficult secondary structure shown below, as well as a GC content that is difficult, and the translated protein has a string of cues. The second case study is one where GENEWIZ was able to synthesize a gene with a poly-A tract. Here the project in question had 30 poly-As in a row. The total gene length in this case was greater than 7,000 base pairs. As you can see, the sequencing data shown below that we were able to synthesize this with 100% accuracy. As far as case study 3, this is to show that GENEWIZ can synthesize large constructs. The construct in question here is a 53,476 base pair construct. On the right hand side, you can see a picture of the gel in which this construct was ran on in lane 3. When digested by pack 1, you can see that the largest band is the 50 kb band for this large construct that was delivered by GENEWIZ. The fourth case study here is a difficult plant sequence. The sequence length was approximately 2,700 base pairs, not very large. However, in this stretch, the GC content was on an average at 37% over a highly variable region, which is throughout the length of the 2,700 base pairs. There were 167 direct repeats, as well as 24 inverted repeats and 10 palindromes in this sequence. Of note, GENEWIZ was able to deliver this final construct within 4 weeks. Overall, our GENEWIZ quality system has project management that plays a role in the initiation of the project by the end user. The project inquiries that come in have a response from our project management team within 24 hours. That is our GENEWIZ guarantee. This is followed by evaluating the project and its design that is provided to us by the customer. At this stage, our specialists do offer comprehensive assessment and recommendations based on their expertise. Some of the other steps include biosecurity screening, as well as ensuring that there is a competitive pricing and turnaround time that we provide to our customers. Once this stage is finished, we move forward with the customers confirming projects. Upon confirmation, the next few steps involve the gene optimization, when the end user requests codon optimization, our team performs that. This helps ensure elimination of undesired structure and making sure that the end product is going to have the best codon bias requested by the customer. Gene synthesis is the next stage, where the synthesis of the desired fragments occur, followed by cloning into the chosen vector, which can either be the PUC57 in-house backbone provided by GENEWIZ at no additional cost, or a custom cloning step. This is followed by quality control, where our team ensures that there's 100% sequence accuracy to what was provided by the customer, as well as a qualitative test of a restriction digest, or a PCR, to ensure that the sequences we deliver are correct. The final stages are product delivery, where the customer receives a certificate of analysis, as well as the final deliverables as requested. These deliverables can be tracked using the information provided by our dedicated sample management team within project management. As you can see on this slide, GENEWIZ project management does provide support from pre-sales till delivery, and usually any questions after delivery as well.
Thank you, Krithika. At this time, we would like to open the floor up for some questions. As a reminder, if you have any questions, please type them into the chat and we will try to address as many as we can. Okay, we got the first question just came in. Can GENEWIZ deliver my gene in my own vector? And can I specify cloning requirements?
Yes, absolutely. You may provide the vector of your interest and a full reference sequence of this vector and specify the cloning requirements, such as the restriction enzymes or any additional strategies. Please note that you would also need to provide us the starting material of the empty vector that we would use for downstream cloning.
Okay, thank you. Okay, let's see. All right, we have another one that just came in. Is there a size limit on the genes I order?
No, and any of our standard gene synthesis services, you can provide us the sequence of any size. However, for the turbo gene that I talked about previously, we do have size restrictions. As I mentioned previously, for the turbo 5, the sequence would have to be minimally complex and under 1.2 kb. And for the turbo gene 7, the sequence would have to be under 2 kb and minimally complex.
Okay, all right, we have another question. This one says, I only have the protein sequence for the gene I would like to order. Can GENEWIZ synthesize my gene?
Yes, absolutely. You may provide the protein sequence on our online order form, choosing the amino acid option. Please also note that you can choose optimization at this step to let us know which nucleotide sequence would be best suited for your downstream needs.
Okay, great, thank you. Let's see, we have another one. Can GENEWIZ ship outside the US, and how much does that cost?
Yes, GENEWIZ is a global company, and we do deliver our products globally. Our shipping charges are $35 for international shipment. This is without dry ice, so any room temperature shipment. As for dry ice, we would have additional charges.
Okay, great, thank you. Let's see, I think we have time for one more question. Will the genes I order be 100% sequence verified? And will you provide any documentation for this?
Yes, all gene synthesis products are 100% sequence verified. Our project team will provide the trace files for the sequencing as well as an alignment file. If the customer requests, we can also provide PDF of the alignment files.
Okay, great. Thank you so much. Okay, well, that's about all the time we have for this session. If your question was not addressed during today's webinar, or if you have any additional questions for Dr. Vaidyanathan, please email us at pm@genewiz.com. You can also visit genewiz.com to see our gene synthesis frequently asked questions. Thank you again for joining today's webinar.